So I think with that, we'll get started. So hi, everyone, and welcome to the TIA Centre Seminar. So my name is Adam Shepherd, and I'm a research fellow here at the University of Warwick. For those of you that are new to our seminars, in these seminars, we aim to invite researchers from across the globe to present new and exciting work. So today, I'd like to introduce Dr. André Homeyer. André is a principal scientist at Fraunhofer Mewis, Germany. He received a PhD in computer science from the University of Groningen. And since 2008, he has been conducting research in the field of computational pathology. His research interests include applications of machine learning and data analytics in biomedicine, with a current focus on technology transfer. So the title of André's talk today is Artificial Intelligence in Pathology, from Prototype to Product. André, it's great to have you here. So just wanted to double check again that it's okay if we ask questions throughout the presentation.
Sure.
Right, and we're also planning to record this talk and put it on YouTube. So is that still okay? Fantastic. Yeah. So with that, if you're happy to get started, please feel free.
Yeah. Thank you, Adam, for the invitation, and I'm happy to present to you some of the work that we've conducted in the Empire project, but more about that later. As you've probably heard in previous talks in the seminar series, there's a wide variety of applications of AI in pathology. By automating common diagnostic tasks like detecting cells or finding rare tissue patterns, AI can help to alleviate the growing shortage of pathologists and improve the accuracy of pathological diagnoses. AI has also shown great potential for extracting novel computational biomarkers from pathology images for precision medicine. The great potential of AI in pathology has caused a surge in research. Here in the slide, you can see a PubMed query for the terms artificial intelligence or machine learning and pathology. And this query resulted in thousands of publications just after the year 2017. However, only very tiny fractions of the prototypes described in these studies have been turned into commercial products for real-world use. Nowadays, there are so-called AutoML tools that automatically generate suitable AI models for a given dataset. This slide shows the result of a study in which we evaluated two popular AutoML tools for applications in pathology. We used these tools to reproduce 3 previously published studies on common use cases in computational pathology. The columns on this slide represent the different use cases: tissue classification here, mutation prediction here, and Gleason grading here in the 3rd column. The crosses represent the results obtained by models of autogluon, and the circles represent results of models obtained by the AutoML framework, AutoML Vision of Google, and the boxes represent the results of the respective original studies by Codray et al. In 2018, Cather et al. 2019, and Avaniti 2018. Here you can see the respective results, the area under the RSC curves, or the Cohen's kappa values. Apparently, the results of the automatically generated AI models are mostly in a similar range, and sometimes even better than the results of the original models, which were created manually. This example shows that the technical implementation of AI models is not so much a challenge than it used to be. However, additional hurdles must be overcome before prototypes of AI solutions in pathology can be turned into products. To be usable in clinical practice, AI solutions must be integrated into the laboratory IT infrastructure. AI solutions are medical devices, therefore regulatory approval must be obtained for their marketing. And to survive in the market, you need a profitable business model. At the same time, pathologists need reimbursement options to cover the cost of using AI solutions. The Empire project, funded by the German Federal Ministry of Economic Affairs and Climate Action, carries out several measures to help AI companies overcome these hurdles. The Empire consortium is coordinated by Charité University Hospital in Berlin, Fraunhofer Mewis, my employer. We are one of the partners in the Empire project, and other partners are the Technical University of Berlin and the Quality Assurance Initiative in Pathology. The rest of this talk is based on publications of the Empire project. Before commercialization, it must first be determined whether a prototype is technically ready for practical use. Technology readiness level are a common way to classify technology readiness. Technology readiness level originate from the aerospace industry. They cover a spectrum from an initial idea to a solution for productive use. And depending on the readiness level of a prototype, the prototype must be further developed or evaluated to a greater or lesser extent before it can be commercialized. Therefore, these levels are a practical measure for internal product management and for communication with investors. We have adapted technology readiness levels to AI solutions in pathology. According to the system, a typical research prototype equals level 3 or 4, depending on the scope of its evaluation. A prototype with demonstrated clinical utility equals level 7, and the final product with regulatory approval equals level 9. To be usable in clinical practice, AI solutions must be integrated into the laboratory IT infrastructure. In pathology, the most important types of software are the image archive, which manages and provides access to slide images, the laboratory information system, which manages all information about the histological workflow, and the pathology workstation. This is the software that the pathologist interacts with most. The pathology workstation provides functionality for exploring and annotating slide images, for accessing patient information, and for submitting reports. AI solutions are mostly directly integrated into the pathology workstation. There are standardized interfaces and established interfaces for accessing the image archive, like DICOM, for example, and for accessing laboratory information system, like HL7 FHIR, the HL7 FHIR standard. However, every pathology workstation has its own proprietary interface for integrating AI solutions. This makes integrating AI solutions into multiple workstations very expensive, especially for small AI solution vendors. To counter this problem, the Empire project developed the Empire app interface. The app interface is an open standard for the integration of pathology AI apps from different vendors into pathology software systems from different vendors. The app interface relies on widely used communication protocols and containerization. To make it as compatible as possible, the app interface was developed considering multiple existing interfaces and feedback from multiple AI vendors. So far, more than 10 commercial apps of different vendors have already been adapted to the interface. Further information can be found in this publication, which also refers to comprehensive online documentation. There are two main ways to integrate AI solutions into pathology workstations with individual advantages. When AI solutions are implemented as workstation extensions, they usually implement a given software interface of their host application for data exchange and user interaction. When AI solutions are implemented as standalone tools, they usually must implement their own data exchange functionality and user interface. The major advantage of workstation extensions is workflow efficiency, because the user can usually operate multiple AI tools from different vendors in one familiar environment. Also, implementing workstation extensions can lower the development effort because one can use existing host functionality. This is of particular relevance in pathology, as implementing functionality to access the multitude of proprietary host-led image file formats can be very cumbersome in practice. On the other hand, standalone tools can offer more flexibility in interaction, parameterization, and visualization because they are not restricted to a particular interface. For the same reason, they are also often better suited for long-running analysis methods with multiple analysis steps that are not supported by all host interfaces. Implementing standalone tools can also be beneficial from a regulatory perspective, as the tools can be certified independently of their host application.
So, André, do you mind if I just ask a question?
Sure.
So in terms of what these workplace extensions, so how do you envisage this to work with algorithms that require GPUs? Is it a case of the pathologist, the lab has to have GPUs available to them, or?
Yeah, workstation extension in this context means that you as a solution developer implement an interface specified by the workstation developer, while a standalone tool would usually be a tool with its own user interface and its own ways of accessing data from the image archive and the laboratory information system. How GPUs are accessed is not really dependent on one implementation options or the other. So we didn't consider this in this regard, or maybe I didn't understand your question completely. Could you maybe specify?
I was just referring, so yeah. I mean, a lot of algorithms that we use for whole cell images require GPUs, right, to run. So I suppose my understanding of the workstation extension was, or having some sort of app that you download, or you, I was wondering how that would, well, use, if it requires GPUs, is it a case of that station needs to have a GPU?
Not necessarily. It really depends on the on this particular solution. The Empire app interface, for example, enables both workstation extensions and standalone tools. In its first version, it was focused very much on allowing workstation extensions, but we figured that standalone tools are very important for practical reasons, especially to enable or to facilitate obtaining regulatory approval, because you can really do all the required tests and documentation for just an individual tool, and you don't have to consider so much the combination of tools. And in practice, a workstation extension, when you use the Empire app interface, is just a container that either implements the analysis method itself and uses GPU resources or general computational resources from the computer it is run on, or which conducts communication with some web services that can run anywhere in the cloud. So it doesn't really matter where the GPUs are and whether an AI solution uses GPUs in the decision of whether to implement it as a workstation extension or a standalone tool.
Okay, thank you.
AI solutions for pathology are in vitro diagnostic medical devices, EVDMDs, because they perform diagnostic tasks outside the human body. As medical devices, they may only be distributed with regulatory approval. This slide shows a greatly simplified overview of a typical approval process. Regulatory requirements vary between countries. In the USA, medical devices are regulated by the Federal Drug Administration. In the European Union, they are regulated by independent organizations, so-called notified bodies. In some countries, regulatory effort can be reduced by showing equivalence with an existing product. But in most countries, obtaining regulatory approval requires similar proofs. First, you need to be certified to work according to a standardized quality management system. You need to be able to provide technical documentation of software requirements, the architecture, and software tests. And very important, you need to conduct a performance evaluation in order to demonstrate scientific validity, analytical performance, and clinical performance. Last but not least, you need to have a plan for post-market surveillance. Currently, multiple countries are working on specific guidelines for AI solutions, for instance, in order to account for their ability to learn continuously. Carrying out a meaningful performance evaluation requires appropriate test datasets. However, compiling such test datasets is very difficult. On the one hand, it is difficult to adequately cover the multiple dimensions of variability of pathology images. For example, tissue morphology is greatly influenced by disease variants and biological factors, genetic, epigenetic, proteomic, that differ between patients, as well as demographic and ethnic groups. And also image appearance is heavily influenced by technical factors like section processing, staining procedure, and scanning. Also, it is very difficult to objectively draw a line between suitable images and unsuitable images that are ambiguous or of insufficient quality for the AI solution to operate correctly. Despite, or maybe because of these difficulties, the current regulatory requirements for test datasets are very vague. In both the European Union and the United States, the regulatory requirements for the evaluation of AI solutions in pathology are still new or are still under development. In 2021, the European Union released a proposal on rules for the evaluation of general AI solutions, so not specific to medicine. And in this proposal, they also impose certain requirements for test datasets. For example, test datasets are supposed to be relevant, representative, free of errors, and complete. They should have the appropriate statistical properties, and they should be subject to appropriate data governance and management practices with regard to design choices, suitability assessment, data collection, and identification of shortcomings. Also in 2021, the FDA defined specific requirements for evaluating AI solutions in pathology in a letter related to the first approval of a commercial product. And the FDA is a little bit more specific, but not much. It requires test datasets to contain an appropriate number of images, whatever appropriate means. It requires them to be representative of the entire spectrum of challenging cases in its intended use. They should cover multiple operators, slide scanners, and clinical sites, and they should be, that's interesting, they should be stratified into relevant subsets, for example, by medical characteristics, patient demographics, and scanning equipment. In general, there is a lack of literature and guidelines for compiling test data sets for AI solutions in pathology. And yeah, as just stated before, also the regulatory requirements do not really help a lot because they are too vague. Therefore,
sorry, André.
Yeah, sorry.
Question from Nasir in the chat. Nasir asked, does representative include people from different ethnic backgrounds too?
Representative means a lot. So there are so many dimensions of variability: scanners, people, ethnicities, different disease subvariants. And I think it's the biggest challenge so far that we face in order to really say whether an AI solution is fit for practice to somehow define what representativeness really means. I think that's really a big open question. Not only what representativeness means, but also where this whole target population of images of an AI solutions, that is the total population of images that an AI solution is supposed to work with in its intended use, where it is, where it ends. So just this, because it's really very subjective to say this image is still good enough for it to be, and I would expect this AI solution to work on. Our recommendation would be to work on automated tools that either accept or reject images based on their quality to make this process a little bit more objective. But as you know, this is still work in progress and a big problem, scientific problem in itself.
Brilliant, thank you.
Yeah, so there's a huge lack of literature. There's so many open questions about compiling test data sets for AI solutions in pathology. And therefore, the Empire project organized a committee to develop recommendations for the creation of test data sets. This committee consisted of various stakeholders, including commercial AI developers, pathologists, and also researchers. And over the course of the year, so mostly 2022, the committee conducted extensive literature reviews and discussions based on their own individual experience in order to develop recommendations on compiling test data sets for evaluating artificial intelligence solutions in pathology. And the results are summarized in the publication shown on this slide, in case you're interested. This is an overview of the most basic recommendations given in the paper. Great simplification, but this is what we think is most essential. First of all, we think it's important to include all images during the routine lab workflow without selection. The best way to do this is to prospectively consider all suitable cases that occur over a given time, like what we've often read is a year, for example, just as a general value of orientation. Retrospective data sets should only be used if there's a clear documentation of the acquisition protocol so that potential biases in the data sets can be excluded. Then it is important to include multiple international laboratories and multiple observers. Laboratories differ in their spectra of patients, diseases, of technical equipment and operating procedures, and also humans have different subjective biases. Then one should estimate the required sample size based on confidence intervals. And test data sets are always samples from the real-world population of images, and that is the total amount of images that an AI solution is supposed to work on. And any performance metric computed on these test data sets is therefore subject to sampling error. The minimum sample size required for a desired level of confidence in the performance metric should be determined prior to data collection. This is not easy, but there are different statistical approaches for doing this for different sample statistics. It's also important to reject images outside the intended use as objectively impossible. That's what I just meant, because images outside the intended use, unsuitable images, should not be included in test data sets because they would heavily distort performance measurements. But since the manual selection of unsuitable images is subjective, it may be useful to use automated quality control software solutions, and hopefully they will become more and more robust and better and generic in the future. We also think that it's important to ensure sufficient independence from development data, because to detect problems with overfitting or shortcut learning, test data sets must be independent from the, it's very important that they are independent from the data sets used in the development of AI solutions. It is not simply enough to use other images, but test data sets should originate from different clinical sites. There are several studies that show that AI methods are prone to exploiting site-specific feature correlations in datasets. And in this case, performance metrics on a test data set that comes from the same clinical site may overestimate the performance in the real world. Just as I've just shown, as required by the FDA, it's important to identify relevant subsets, because AI solutions that are very accurate on average often perform much worse on certain subsets of images. For example, such subsets may consist of certain disease variants, demographic groups, or also a particular scanning equipment, for example. To ensure that AI solutions work well in general, they must be evaluated independently on relevant subsets. Identifying subsets is also not that easy, but there are, for example, usually this has to be done on the basis of world knowledge, but as an aid, it's also possible to use unsupervised clustering approaches to identify potential subsets automatically. And this was a very controversial recommendation that we reached in the paper, and that is that for test data sets, you should avoid using synthetic data. In recent years, it has become common to augment training data sets for AI methods using synthetically generated image data, which is good and fine and very helpful. But we think that such methods are unsuitable to generate test data. Such methods always rely on some simplified model of real-world variability, and therefore are likely to introduce biases in a test data set, which make meaningful performance measurement impossible. Successful commercialization of AI solutions for pathology requires a sustainable business model. A popular approach to developing business models are so-called business model canvases, which you can see here on the slide. This is an exemplary business model canvas for AI solutions in pathology. At the heart of every business model canvas is the value proposition for customers. For AI solutions in pathology, the principal value propositions are cost reduction, that is assisting pathologists to perform more work in less time, quality improvement, that is improving the accuracy, sensitivity, or reproducibility of diagnostic tests, and also innovation, that is extracting innovative computational biomarkers for precision medicine from standard pathology slides. The right side of the business model canvas focuses on customer-related aspects, such as customer relationships, customer segments, sales channels, and revenue streams. And the left side of the canvas focuses on business-related aspects, key partners, key activities, key resources, and their cost structure. A detailed discussion of these aspects is out of the scope of this presentation, but I would like to refer to this publication cited below, because in this publication we treat these aspects in more detail. And what's very important is to check the assumptions made in a business model canvas very early and continuously during the development of AI solutions. For pathology, yeah, Adam, sorry.
Questions from the chat. Yeah, sorry, just a couple of questions from the chat here from Nasir. So Nasir's asked two different questions relating to probably the slide before that one. So the first one was, does the FDA have any requirements at all on minimum accuracy or sensitivity or specificity, for instance?
Not that I'm aware of. Generally, you won't find any numbers in these recommendations, no 0.9 something or anything like that. Or what would also be great is if the FDA or the EU said how many images even a certain dimension of images that you should consider for evaluating AI solutions. Practically, obviously, that wouldn't be reasonable, because the number of images that you need really depends on the variability of your data set or your target population, and also the confidence and the precision you want to have for your performance metrics. And it's all very complicated. And yeah, as stated on this slide, the regulatory requirements are very vague. They are more specific in the US than they are in the European Union, and far more advanced as well in the US, especially because of this this first cleared product for marketing in 2021, but still, it's really not very helpful if you're a small AI solution developer and you wonder how to conduct your evaluation.
No, thank you. And we've got a second question here. It was from Nasir again. He said, asked, from a regulatory point of view, do you see any future in the use of synthetic data for training, pre-training, and fine-tuning? I know you've touched a bit on that in one of your latest slides.
Training, yes. So this point did not refer to using synthetic data in the development of AI solution. This just referred to the use of synthetic data in the testing of AI solutions. So I think synthetic data is always based on certain assumptions that you make about the real world, or the data set that you fed into the method that generated this synthetic data. So it's never able to cover the entire variability, and will always have certain biases. And that's why we recommend not to avoid using synthetic data for testing purposes.
That makes sense. Thank
but
you.
we would definitely recommend it for training purposes. Yeah. Okay, are there any additional questions?
No, I think that's all the questions so far. So thank you very much.
Okay. For pathology labs, the use of AI solutions causes investment and operating costs. In this illustration here, these are the pathology labs. And to make the use of, because the pathology labs have to pay the solution developers for using the AI solutions, licensing costs, whatever, and they also need to acquire the necessary equipment, like GPUs, for example. To make the use economically viable, costs must be reimbursable from either health insurance companies or from the patients themselves. Billing medical services, unfortunately, is extremely complex and varies between different countries. More details, again, are given in this publication. However, most medical billing systems rely on a so-called fee schedule that lists the content and fees of various medical services. For instance, in the USA, fees paid by the Medicare insurance program are based on the Medicare physician fee schedule, the MPFS, or the clinical laboratory fee schedule, CLFS, for clinical laboratory services. In Germany, there are two different fee schedules. They are the so-called Einheitlicher Bewertungsmaßstab, very complicated word, which is used by the German statutory insurance entities, and the Gebührenordnung für Ärzte, which is used by the private insurance entities. Both fee schedules work a little differently, and also the codes in there are not really comparable. It's very complicated topic to understand, especially for small startups. When you as a startup want to introduce new codes in a fee schedule for your AI solutions, you need to involve a lot of different stakeholder groups. And in particular, a particular important role is played by the associations of pathologists, such as the College of American Pathologists in the US or the Bundesverband Deutscher Pathologen in Germany. In order to introduce new codes, it's essential to demonstrate the clinical utility of the AI solution. For example, you need to demonstrate that the use of the AI solution really improves in some indirect manner patient outcome or the cost-benefit ratio. For example, that you can make the whole treatment more economical. Unfortunately, today there are no general listings for AI-based computer assistants in pathology in the fee schedules in the USA or in Germany. However, additional reimbursement is already available for certain computer assisted diagnostic procedures. In the US, it is possible to obtain reimbursement for the use of computer assistants in the analysis of cytology specimens for cervical cancer screening. The amount reimbursable is between $6 and $16 per case, and the reason is that the value of computer assistance is thought to lie in reducing screening errors and increasing sensitivity. Also in the USA, it is possible to obtain reimbursement for using computer assistance in the assessment of immunohistochemistry, biomarkers like ERPR, PD-L1, Ki67, or HER2. The value here of computer support is to improve the accuracy and reproducibility of the resulting scores. It's interesting to note that the the possibility to obtain reimbursement exists for computer support exists already since 2004, so it's very old. However, the additional fees for computer support declined from about $30 in 2009 to $1.80 in 2020. It's not really a lot that you can charge for a computer system in this regard, although this is probably one of the most important applications. In Germany, unfortunately, there is only one single diagnostic procedure for which computer assistance can be reimbursed, and this is called, it's a little obscure technique, it's called DNA image cytometry. this procedure can be used to grade prostate tumors based on irregularities in the DNA content of cells. Computer assistance is claimed to enable the analysis of more cells and therefore higher sensitivity. And this computer assistance can be billed to the patient on a self-pay basis at an amount of 162 euros. Quite a lot, obviously, especially compared to the rates in the US, but we were not able to find any figures on how often this really occurs. So how many patients are really ready to pay this additional heavy charge for having a little bit more accurate assessment of their tissue sections. In summary, unfortunately, there are still big challenges to bring AI solutions for pathology to the market, but these challenges are best met through collaboration. It's very important to, first of all, it's very important to collaborate with pathologists. You should, as a small startup, you should always create partnerships with pathologists, and you should give them incentives for data sharing and for consulting and whatever. It's very important to cooperate with multiple laboratories to cover the respective variabilities and to get input from different sites. And we think it's also important to provide pathologists early with prototypes and to obtain their feedback in order to develop solutions that are actually usable in practice. We would also recommend to collaborate with workstation manufacturers, and you could, especially when you're very small, you could enter strategic partnerships with them and integrate your AI solutions into their workstations. In this manner, you can benefit from their existing sales channels and also their customers. Last but not least, we think it's very important to work with the pathology associations. As stated before, this is crucial for establishing reimbursement options and also for shaping technical standards. And in particular, we would recommend to participate in dedicated digital pathology organizations like the DPA or the SDIP, because this is really the venue where all the relevant experts are involved. This was my small, brief, concise presentation. And in case you want to know more, I would like to refer to this publication, which was very often cited on many slides and provides some more input, some more information. And yeah, thank you very much for your attention. And I'm happy to answer or try to answer any remaining questions.
Brilliant. Thank you for a fantastic talk, André. So I'll open the floor now for questions. I think, Mustafa, you've got a question?
Yes, thank you, André. Really good talk. So going back to the matter of using synthetic images, I'm talking about the test time utilization of them, okay? But do you think they can be actually used to benchmark some of the domain shift or variances? I mean, the performance of some of the domain shifts that are known in computational pathology. For example, if you want to benchmark a data set, an algorithm or a product on how much does it do well on the changing of illumination or staining or focus deterioration. So how much does it can endure this kind of domain shifts or artifacts, right? So with real world data, you cannot really control or measure that. Whereas with synthetically generated data, you can actually benchmark that, right? So you can, for example, have a certain data set, adding small bits of, blur to that images and calculate how well the product perform until which, level of distortion. So this kind of use cases of synthetic data, I think they can be used. What do you think?
Yeah, I think this can definitely be helpful in assessing the practical value of a solution, but it's I think it's no replacement for having a really good, very comprehensive test data set from the real world. It's a good supplement, I would say, but you still need this other data set that really contains real cases from the real world with a very high variability in order to assess practical applicability.
Obviously, yeah, but like in terms of having, I mean, I would think it would be good to have another section of like predefined robustness analysis, which you can like examine the robustness of the products on certain problems. But you're right, they cannot be used as the main testing set. I completely agree. But I don't think we should avoid them at all.
Yeah, good point. Our paper was really concerned with a data set that tries to tell you how well, that tries to produce a performance metric that really tells you how well your solution works in the real world. And as I said, it's very important to also consider different subsets of this real world data set, and maybe synthetic data could become handy in evaluating certain aspects of the general real world performance.
Yeah, true. Thank you.
Brilliant, thank you. Do we have any other questions from anyone in the chat at all? No. Okay, so I suppose, I mean, in terms of algorithms, what do you think is the main limitation or thing holding back a lot of different algorithms from actually being implemented? I know obviously you've just described a lot in terms of, how do I describe? So what I'm trying to get at really is sort of the explainability and lack of interpretability of some deep learning methods. So how do you think we can try and get some of these methods into the clinic? Is it a case of we need to make our methods more transparent, or
very good questions. Actually, some colleagues from the Empire project conducted a study on explainability, where they conducted a survey with a lot of pathologists, where they presented different kinds of explainability methods to them and tried to assess their opinion on them. And turned out that all of these explainability methods are very complicated to use in practice, because you all know how strained our health systems are in every country, and I think we really need to make healthcare cheaper, more efficient. And when you explainability or testing explainability will always introduce some further effort, which reduces the efficiency of the whole pathological assessment process. And maybe if this is too much effort, then AI is no longer economically worthwhile. From my point of view, I think we really the main point what's holding AI back at the moment is really not the AI itself, but it's the lack of digitization in pathology labs. That's what we all also realize in the Empire project. So many of the project partners in the Empire project are working in this area for quite a long time, and we know about the state of digitization in pathology labs in Europe. And we talked to new labs that we had not talked to before, and we were really horrified how difficult it is in practice to digitize them and to give them economical incentives to work digitally. I think this is the biggest aspect of the biggest barrier to adoption of AI. And also, I think the economic value of AI currently is still not big enough. So the real advantage that you have by using AI. So most of these solutions that are economically available, the real benefit is an improvement of quality and not really an improvement of efficiency. And I think we really need to work on improving the general workflow efficiency in pathology to to make it economically worthwhile.
Thank you. And the Nasir said in the chat, I'm sure we can use AI to make healthcare great again. And also going back to one of your previous slides, actually. So I know it was about computer assisted tools, but how come do you think that the price of the reimbursement prices, well, not prices, the reimbursement amounts had reduced over the last 20 years, whereas you'd sort of expect as technology has improved that those sort of values would increase?
Actually, we were also surprised that by the amount that especially the rates for immunohistochemistry declined over the years. And I think that I'm not completely aware of the whole story behind this reimbursement option, but I think there was a company which lobbied strongly for introducing this reimbursement opportunity in 2004. I think the overall quality at that time, long before the age of AI, was not really good enough to have a big impact on value in practice. And maybe this is the reason why it declined from year to year. But I'm not really, I'm no expert on why it declined so much and by how much. I don't know. Sorry, did this answer your question? Or
no, that answers my question. Thank you. Okay, so if we don't have any more, just see if we have any more questions from the group at all. If not, I suppose we can end it here. But thank you so much for your really interesting talk today, and we wish you the best of luck with your future work. And I'd also like to say that we've got another talk again in 2 weeks' time on Monday, the 6th of February, where we'll be joined by Sarah Fremont from Leiden University in the Netherlands. So again, thank you very much for a really interesting talk, André. And yeah, hopefully see everyone again in 2 weeks' time.
Thank you, Adam, and all the best to you as well.
Thank you very much.
Thank you. Bye.